Thank you for joining us this afternoon. My name is Dr. Emily Sena. I'm a senior lecturer at the University of Edinburgh, and I'm very happy to see lots of people join us for the second of our 3 in vivo webinars that the BNA is running as part of our work to strengthen the credibility in neuroscience. And we're very grateful for the support given to this campaign by the Gatsby Foundation. A few housekeeping things. Can we ask people to put their questions into the Q&A tab in Zoom, and then use the chat function for general conversation. If you put your questions in chat, they may get missed. Also, the webinar has been recorded and will be made available on the BNA's YouTube channel at the end. So the format of these bite-sized lunchtime webinars is compact. We only have 30 minutes. The way it will work is that Natasha will speak for about 15 minutes, and then the rest, I think we'll have another 15 minutes, so the rest of the half an hour will be available for questions. So get your questions in early to make sure that we get to them. I'm not going to talk too much. I'm just going to quickly introduce Natasha. I've known Natasha for many years. I'm delighted that you all have the opportunity to hear her speak about this, because she's knowledgeable and interesting and very compelling in her talks. So for those of you that don't know Natasha, Natasha Karp's the director in AstraZeneca, and she leads a team of statisticians supporting the preclinical pipeline. She's been in this business now for about 18 years and has worked both in commercial and academic settings. And Natasha says that she's driven to finding practical solutions to the challenges of modern biology, and acts as a bridge between theoretical statisticians and biologists. And I think this is something that's unique to Natasha's skill set, and that's something that she does very well. In recent years, she's focused on the challenges in preclinical research, with a particular interest in improving the replicability, reproducibility, and generalizability of studies. So with that, I'm going to pass on to Natasha. Thank you very much.
So I'm just sharing my screen, hopefully. Okay, so I'm going to touch on 4 different areas. Reflecting on our traditional design, the challenges that arise from phenotypic plasticity, then reflect on how should we introduce variation into our experiments, and the value of multi-batch designs. So when we are conducting experiments, we have to simplify a very complex world into a feasible testing space. We have to make it doable research. We're trying to isolate cause and effect. So the only difference between our control and our intervention group is the intervention, so we can assign causality. So this means we're trying to manage potential confounders, sources of variation, and we choose to standardize them to make them exactly the same. This is to remove the confounding effect, but also has the benefit that it lowers the variability, which can maximize the sensitivity to detect a treatment effect, which means it reduces the N needed. That has a cost benefit, and when we're working with animals, it's ethical and is a driver under our 3R framework. So the outcome of these drivers are experiments that are highly standardized. They're typically one sex, mostly male. We will conduct the experiments typically in one batch in time. We will be often working on one genetic background with one age under one housing and hazarding system. And so we are testing in quite a narrow testing space. And this raises questions about the external validity, the ability to apply these to other populations, the ability to generalize the results. And if you think, my own genetic background, if we duplicate myself and my lifestyle, how applicable are the findings on me to others in the community? And then it raises the question, is this equivalent to clinical trials, where a lot of our research is aiming to inform? And this issue is arising, this generalizability issue is arising from an issue with the concept of phenotypic plasticity. So what do I mean by phenotypic plasticity? So living organisms are highly responsive to the environment, and leads to phenotypic changes, both with long and short-term duration. And in fact, they can even be localized, as we can see by the suntan marks in that photo. And this ability to respond is essential for survival. But that means, and is part of the ability for us to have a treatment effect, because it depends on what we expose something to. So it's inherently a part of the concept of science. So the direction and magnitude of a treatment effect depends not only on the nature, duration, and intensity of that intervention, but also on the animal's or cell's current phenotype and the experimental context. So the history of the animals and how you're running that exactly. So back in 1999, there were a group of scientists working with in vivo animals that were already grappling with issues of reproducibility, and these were in the behavioral fields. And they found that they couldn't reproduce the observations in one lab to another. So a group of scientists came together to study strain effects, so 6 strain effects across 6 behavioral screens across 3 labs. And to try and to reproduce their findings, they conducted extensive standardization, not just of the protocol, but of the housing and husbandry, a common source of animals. And the issue that they found and published was that even with that standardization, they could not reproduce the results. And it wasn't that the results had a different size in different institutes, but actually the direction of the effect could be in opposite directions. So from this, there were 2 calls. Some of the community said the answer is to standardize further. We just have to work out what the animals are reacting to and report that metadata so that it can then be reproduced, giving us replicability. But another set of the community said, actually, there's the standardization fallacy. With every additional variable that is standardized, the testing space narrows, and then we can get idiosyncratic findings to that particular environment, because there is no pure treatment effect. It's not that we're trying to get rid of noise to isolate the treatment effect. The treatment effect is context dependent and should only be interpreted relative to the normal variation. So if we need a paradigm shift in how we conduct experiments, we want to introduce variation. This isn't a free-for-all that we stop standardizing things, because we will lose our sensitivity or introduce confounders. But actually what we need to do is embrace randomized block designs. So what do I mean by a block? So a block is a set of experimental conditions where you have standardized, but between blocks, things are varying. So this could be that it's a different institute, it could be a different operator, it could be a different genetic strain, it could be multiple things coming together. And then when you statistically analyze the data, you account for block and you estimate the average effect across blocks. And there's 2 different ways of designing these experiments shown in the diagram there on the right. In the middle, we've only got 1 animal per treatment group per block. So there's no replication within a block. On the right, we have replication within a block. The replication within a block gives you an advantage that you can estimate the effect within a block and then estimate the average across, but it uses more animals. So it depends on your goals, practical consideration, which design that you do, but both are replicable. The value of embracing multi-block designs of improving reproducibility has been proven, and this paper used simulated experiments using real data from databases of animal data for treatments for either stroke, myocardial infarction, or breast cancer. And for a particular treatment, they conducted a meta-analysis to calculate the true effect. And then they used that data for that treatment to simulate different ways to designing the experiments. So they could either run a single lab study or they could run a multi-lab study, but the total number of animals were run with the same. And they estimated within that single experiment what was the estimated effect in the 95% confidence interval. And if that included the true effect, that was a success. And so they were calculating the coverage probability, how often the true effect was within this 95% confidence interval. And this is what they found, that the coverage probability increased as you increased the number of labs. As you had more labs in your data set, even though the N was exactly the same, you would more reliably detect the true treatment effect. They had a counterintuitive finding that as you increased the N, you were less likely, you had a lower coverage probability. And that was in rising because you're increasing the power to detect a smaller effect that could be idiosyncratic to a certain environment. And this is a way to see it more visually. And so this is some of a subset of the simulated data from those studies. So if we look at the diagram on the left, this is the data from 1 lab. The dotted line is the true effect. The dot and the bar represents the estimated effect and its 95% confidence interval. And what we can see for each the simulated studies in the one lab design is that they're quite scattered around the dotted line. They're rounded, so on average it would be the dotted line, but actually there's some that are a very long way away, positive and negative. There's a lot of scatter. As soon as you start adding more labs into the data set, what happens is that estimate's being pulled tighter to the true effect, and the data is becoming more reliable. But the challenge here is this data was never intended for this purpose. The difference between data set 1, data set 2, data set 3 is immense. It could be a different genetic background. It would be a different source of animals. It will be a different housing and husbandry condition. So the question is, well, how do we embrace this sort of design? Well, which variables will matter? And how much variation do we need to introduce? And that's slightly going to depend on the biology of your particular experiment. So factors have been considered, such as time, morning and afternoon, operator, environmental enrichment, genetics, sex, or the lab. And they've shown some success, but there's no hard and fast rules yet on what we should do. So I would like to propose, and one of the things that I've written about is the potential of multi-batch design. So what's a multi-batch design? That is for your own institute that you run your experiment with smaller batches, individual batches, and then you run so almost like a meta-analysis, accounting for batch to estimate the effect across. With the idea being that you've set your metadata, but even though you've set the metadata, there are some things that are still changing that the animals are responding to, so the readings are going to be higher and lower. But because you're estimating an average effect across the 3 batches, it will be reproducible for your metadata for your conditions. And I'd like to demonstrate where we've used this within AstraZeneca. So in AstraZeneca, we do some syngenic studies. So this is where we've got some genetically identical mice with a genetically identical tumor in the flank. We randomly assign the animals either to a vehicle or treatment group, and we monitor the tumor with time to assess for a treatment effect on tumor growth. Now, these studies are variable. They're variable within studies and between studies. So we were tending to run very large studies multiple times. And what we suggested was then, if we're going to run it multiple times, let's just embrace that idea from the beginning. But instead of running large experiments multiple times, lower the animal number within a batch, because we are intending to bring that data all together in our estimate. And this also means that when you're estimating the effect, you're looking at 3 batches with equal input. You are not cherry picking the data, taking the one that looks good and matches your hypothesis. You are using all the data to estimate your effect. And here is an example of some of the output. So on the left, we've got the growth curves for the 3 batches, and we've calculated the summary metric, the growth rate. And you can see on the diagram on the right that it is variable between batches. It's variable in the vehicle group, how fast the tumors are growing, and the size of the effects for a treatment across the batches. And so, but we can see that they are all in the same general direction and now can estimate an average effect, and they were all significant. But we now have got much greater confidence from this one single experiment. We don't need to reproduce it, and we can then go on to think about other aspects of the research we need to conduct. As part of that research, we did some power calculations through simulations of the data. And we did this for 3 different tumor models. And we were comparing the number of animals we would have needed for a 1-batch approach versus the multi-batch approach for a 30% change in growth rate. When we were conducting the multi-batch approach, even if we hit the power we needed, we always said we had to have a minimum of 3 batches so that we had an estimate across multiple batches. For 4TI tumor, which is a very tightly growing, it's a very consistent tumor model. Actually, the difference in the N was no difference between embracing a 1-batch approach that had been run 3 times and the multi-batch design. For a 2-group comparison, we would need 18 animals in both set of experiments. But for MC38, which is a more variable tumor, when you account for its variation from our historic data, for the 1-batch approach for a single experiment, we would have needed 28 animals for the control and the treated. So if we had replicated it 3 times, we would have used 84 animals. But if we embraced the multi-batch design from the beginning, we would have had 3 batches with 6 animals per group, and we would have used a total of 36 animals. So we could lower the number of animals we used and have a more reproducible estimate and used all our data to make the decisions as we go further in our research. So, traditionally, we are coming from a culture where there has been a call to standardize and leading us to conduct studies within a narrow window of testing space where we are then trying to generalize the results. And I would say that this is leading to us having issues with translation and reproducibility. We do need to start working out how we're going to embrace designs that identify more generalizable results. And the recommendations are block designs. So they introduce that variation in a controlled manner. And I really strongly believe that multi-batch designs is a good starting point, because at least you would know within your lab, with your setup, this is a reliable estimate. And I've put together some links, which hopefully will be provided to you, some papers that you could read if you want to know more about this, and a very informative and effective video that's only 3 minutes long. And I look forward to hearing your questions.
Thank you, Natasha. That was I always learn something new. It's fantastic. I think I will kick off the questions and give people opportunity to, I guess, coalesce their thoughts and put their questions in. So I was interested in your point about how the batch experiments could essentially end up with using fewer animals.
Mm.
And this, people, in using animals in our research, we've got the 3 R's consideration, and this clearly is a reduction issue. And I just wondered if you're aware of engagement from ethical review boards in this space, and if not, how we could, I guess, support that implementation.
No, I don't think I've been aware of engagement in ethical review boards. I think one of the things is that the challenge of working out how to power the experiments. So, there I constructed simulations. Well, that's a skill set that most biologists wouldn't have. And therefore, that's the complexity that makes it harder. There are also practical issues when you're running this sort of experiments in a lab, because when you're instead of ordering a space in a lab at one point in time, it's more complex, the study, because it's 3 sets of animals. You're asking for resources across 3 points in time, and sometimes it can be harder because people want to see the results as you're going, and you go, I haven't got enough data yet. You have to wait to the end of the study to get the data, so it does take slightly longer. So to embrace it, I think we need to tackle the practical issues, which is maybe how to design block designs to enable this. But also, I think the other practical block, I would say, is the analysis. People know how to do a t-test. They know how to visualize the data. GraphPad doesn't actually support block designs. And so that's a common tool that our scientists are using. So therefore, there's an inherent blocker. Internally, we built an interface, a tool that enables scientists to do this readily. But I don't think that's available externally. So those are the areas where we could work to enable scientists, and then it will reduce the activation energy to embracing it.
Essentially, we need more Natashas who speak both biology and statistics. To act as the bridge. Okay, we've had a few questions come in. So the first is from Jordi Lopez Tremoleda, if I pronounced that correctly. Always great. They had a question about multi-batch, thinking it's not always feasible to mix animal injections to different cells, as this would depend on study requirements.
I'm not sure I actually can quite fully pass that sentence. To mix animals injected into different cells. I think it's the same type of cell that I was talking about. It's in this context, it's the same tumor type. It's just that maybe it's the batch of cells or the preparation is different. So that's what I mean. It's just, it's a bit like if you would, for an individual experiment, a large one, you would prepare the cells that you're going to inject and inject them. You would have completely independent preps of cells, because in a way you're wanting to allow that variation to occur. That's what I mean by separate cells. Hopefully that's answered the question.
Yes, okay, agree if these are batches. Okay, I think you have answered the question. Rosie, I think you've mentioned this already, is asking about how you analyze these sorts of batch data.
Yes. So there are a couple of ways that you can analyze this data. And actually in the paper, we compare 3 different ways of analyzing the method. So one is to use a sort of a meta-analysis, so that you're treating each mini experiment and then using the sort of the means and standard deviations from each mini experiments to build up to estimate across the 3. Another option is to treat batch as a fixed effect, and the third option is to treat it as a random effect. And so what do I mean by fixed and what do I mean by random? So intellectually, random means that we don't care particularly about batch, but we just think that batch represents a population. And actually that is what we want. We want it to represent it. That could be, we've got 3 batches, but actually there could be a population of 20. So you include batch as a random term in your model, that it adds variation. So it's called a mixed model analysis. The other option is to treat batch as a fixed effect. So it's a bit like a factorial analysis, but instead of allowing the treatment to interact with batch, because we're not interested in that, we just want to estimate the treatment effect across them, you're adjusting for the batch being higher and lower. There are pros and cons between those different methods. If you've only got a few batches, you could argue that you haven't got enough to estimate across the random effect of it. So it'd be better to account for it as a fixed effect. And that's where statisticians can argue with ourselves for hours on end. So the results weren't particularly different for those data sets. I have a tool that in-house where we process it either way, and the results weren't near enough the same. So does it really matter? So I would suggest the easiest way would be to include batch as a fixed effect to adjust for it and then estimate your treatment effect across the batches. You can do this with the tool called vivostats, which is a point and click interface, but you can't currently do that with graphpad.
And I guess that will be dependent on the magnitude of the effect in terms of because if the effects are relatively big, then these different methods
will have less of a but if you're right on the edge of the border of significance, yes, it can be falling in and out of significance. Exactly. But that's because of how we treat the p-value as a gospel truth, isn't it? So if you step away from that, actually, if it's 0.06 and 0.04, it's not actually that dissimilar. So, and that's always the risk. It's knowing the assumptions. So when we talk about selecting the method, it's like knowing, is it good enough to answer your question, and overall can you justify the assumptions you are making?
Which makes sense. And to be honest, I didn't quite, so my understanding of fixed and random, I'm now realizing maybe wasn't quite correct, because I think my assumption would have been that because you're testing the same thing, that you assume that effect is fixed.
No, fixed
population, because your population is technically the same, even though it's batches.
No, mathematically, I think we should revisit that maybe separately.
But this is my ignorance.
So a fixed effect is something like sex can be a fixed effect. So I care that males and females are different. I want to know what the effect of sex is. Now, genetic strain can be treated as both a fixed effect or a random effect, depending on what you're trying to do. If I care about the difference of this strain versus that one, I would treat it as a fixed effect. But if I've designed my experiment to have 5 different genetic backgrounds, and I want to say, what is my effect across mice in general, I'm treating my strains as a random effect, as from a population of strains. So then I treat it as a random effect. So so there's a paper in the British Medical, in your journal, which goes into this quite well, that I wrote. So if you want to know more, I'd recommend you have a look at that.
Thank you. We've got 2 more questions at the moment. So fascinating talk. How would one go about calculating power? Oh yes, so this is something you mentioned as well, and the required N for a multi-batch study. And is it different from how would normally calculate power for a study?
The answer is yes. Unfortunately, it is different. So when you're calculating statistical power, you're deciding what you're trying to you've got your biological question and your statistical question that you're using to answer that biological question. And then for that statistical test, you're using a set of power tools that hopefully exist. Because you're using a different analysis pipeline, you need a different set of statistical tools. The way that I had done this was using historic data and simulations, and the code is available in the code that I used was available in the papers that I published. So this is R code, and so you could use that code and input different variables to model the effect of power on those sorts of things. But that's not easy because you need to have some experience of programming and working with R. I think it's I do feel very pulled in many directions. It would be something that needs to be worked on.
Yeah.
For the community.
And I guess there's another question here that's asking about, can graphpad be tweaked? But I'm guessing you would say you need to do it in R.
No, I'm saying you can do it in vivostats. So in vivostats actually has R in the background, but you don't have to interact with the R at all. It is all through point and click interfaces. It's a bit like SPSS. So SPSS, the point and click interface, would also do it. That would be fine. In vivostats has the language that I was using. It has a textbook that goes with it that will take you through it. So, it is a strong contender. Potentially you could also use R Commander, which has R Commander is a interface. It's got R in the background, but it shows you the R code that it generates when you touch the button. So it's a tool if you want to transition into R. So you can sort of see how it's generating the code, and then you can slightly tweak it if you want. So working between those two, that is another option. But yeah, graphpad is a very powerful tool for a lot of situations, but it's a real pain when it can't do certain things.
Yeah. So I've got one final question, which relates to the your earlier slides where you talked about multicenter studies. And I guess a lot of conversations that I've had in this space, multicenter studies have been touted as the way to introduce this heterogeneity. And I just, not thinking about pursuing an academic setting and the incentives around collaboration and all that kind of thing, what would you say are the added or not added benefits between the multicenter and multi-batch approach, all other things being equal?
So the multicenter method was proposed quite late in your pipeline. But if we start addressing reproducibility late in the pipeline, that's a bad point to address it. So that's my reservation with that method. If we get so far and then initiate this huge project and then it doesn't reproduce, that's problematic, which is why I think trying to make it more reproduce earlier is a more effective strategy. Certainly it is. A multilab is the similar concept. It's easier to introduce variation because you're, in a key variable for a key treatment, we could, replicate. It could be a good strategy, and particularly in light of, the big findings that are not being reproduced, or there's a paper that's just come out from the cancer area reproducibility project where the effect size is on reproduction is 85% lower. It's hugely lower, and that would have been picked up by a multi-lab study. And certainly clinical trials fail often because of a lack of efficacy. So if we can work that out sooner, that would be better. Yeah,
that makes sense. Well, thank you. We are just about at time. I saw a comment that was requesting you to come back and explain statistics for dummies, and I think that's testament to how well you're able to articulate some of these complicated concepts to those of us that don't have those expertise or background. So thank you very much.
Thank you for the feedback.
What was that, sorry?
Thank you for the feedback.
Yeah, no, it's I know I say it all the time, but it's very true. So we have one more in the series, in vivo webinar series, that's coming up next Wednesday. So it's on a Wednesday on the 16th with Nadia Solomon from Imperial. So it'd be great to see as many of you as possible there to hear what she has to say. I also want to give a quick plug to the Credibility Prize. So the BNA Credibility Prize is the second year the prize is running. Nominations for the prize close next Tuesday, on Tuesday the 15th. I highly recommend people to consider nominating colleagues that they think are deserving of a prize. There's 3 prize categories: early career, individual and team prizes. You can have a look at the BNA website. It shows who won the prizes last year. If you're a member of the BNA, you can self-nominate as well, but you are able to, BNA members are able to nominate people who aren't members. So have a think about that. And if you have any questions about the prizes, then please do get in touch. Great. With that, I will let you go. I'll say thank you once again to Natasha for your time and sharing your knowledge. And thank you all for joining us this afternoon.